you don't know how wonderful it is for a teacher to be able to introduce a student who's done incredible things. Okay? And Erin is a Smithy. Okay, biochemistry major. And as we were talking about last night, she was actually, in some ways, she and her cohort were a guinea pig in that she took Biochemistry 1 the first time it was ever taught. Biochemistry was only a one semester course before. We split it into two, and I got to teach the first semester. And she was there. She took a couple of other courses too. She did a fabulous honors thesis, which I was her reader on because she did the thesis actually at the National Magnet Lab that was then in Cambridge. And did some fabulous work on insulin structure using NMR. Back then it was new, really new. Some of the techniques that she was talking to me about, I didn't know at all, okay? And I guess the reason I didn't know them is that they were just being developed. And so I said to Erin, well, before I can read your thesis and comment on it, could you explain the techniques, please? And she did. It was fabulous. So she was clearly a born teacher, too. Left Smith, went on to MIT, got her PhD in 2.5 years. Okay.
2.5 years.
Yeah. Then went on, did a postdoc out in California. While she was in her postdoc, was offered a position at the University of California, San Francisco. So stayed there for quite a few years until he was spirited away to Harvard and the Broad Institute and other such wonderful things. And then in 2000 became a Howard Hughes Medical Institute investigator. Then started to move into administration while keeping her lab, and became then the president of the Howard Hughes Medical Institute. First woman to do that, so she broke a ceiling there. One of the youngest women ever inducted into the National Academy of Science. And a very devoted Smith alumna. The one thing I really thank her for is whenever I ask her for something involving Smith, she says, when? I'll be there. Loyal. So, I've spent enough time talking about Erin. Erin O'Shea.
Hey. All right. Thank you. Thanks, Mr. Skordillis, as we used to say. Now I have to say Stan, but it's hard to get over the old habits. But seriously, I'm really, really happy to be here. You know, Smith really changed my life and had an enormous influence on my career. And before I get into the science, I just want to say a few words about that. I was a student. I graduated almost 30 years ago now. And let me tell you, it may not seem this way now, but time flies after you graduate. And, you know, once you start in on your career and everything, you know, treasure the time you have here because it's really something special. But yeah, I graduated in 1988. As Mr. Skordillis said, with a degree in biochemistry, and I got a huge amount out of being here. I developed a sort of confidence and belief that I could really do anything that I set out to do. And that's something that is really powerful. And it came from my experiences here. It came from people like Mr. Skordillis, you know, really encouraging me and supporting me the whole way. I also just got a lot out of my fellow classmates here. I mean, it's something very special, I think you all know, to be at a women's college, and especially as a scientist when, you know, I was in a field that was dominated by men, and again, this sort of confidence that I got from being here and the belief that I could really do anything that I set my mind to was something very, very important that I got out of Smith. And on top of all of that, I got a tremendous education in science here. Other things too, but for my career as a scientist, I got a terrific education in science. And I don't just mean general, you know, science education. Of course, I got that, mathematics, physics, chemistry, biology. But I took a class with Mr. Skordillis where I learned some material about muscle proteins, which you all know he's passionate about, that really launched my career. And it's the reason why I finished graduate school in 2.5 years. I learned some things about muscle proteins that prepared me to make an important discovery about transcription factors. And that came directly from this class. So I guess my message there is that soak it all up, because you never know when information you learn might be useful in another context. And for me, that information about these coiled coil proteins involved in muscle, you know, I took that information and was able to see things about a problem that other people didn't see. And it was solely because I had this background coming from a class here at Smith. So, yeah, and I mean really important. I had 2 papers in science in 4 months as a beginning graduate student. And then that's why I graduated so quickly. But it really was this insight coming from the class. So I got a lot out of Smith. You guys are all lucky to be here and enjoying the time. And so now what I want to do is to just tell you about some work from my lab. This is a project. It's really a series of projects that's been carried on almost exclusively by PhD students in my lab, with some participation from undergraduates, but not so much the postdoctoral fellows. So these are people, you know, in graduate school, and as you'll hear, they've done a bunch of pretty interesting projects. And I want to use this as sort of a way to explain how we decide what questions to ask, and then how we actually answer the questions. And this peculiar system I'll tell you about makes a kind of good case study for those types of issues. Okay, so I'm going to talk to you about the circadian clock, and first I need to tell you what that is. So circadian clocks are timekeeping mechanisms that almost all cells, particularly cells in animals, have. Okay, so I mean, this is, when you think about it, this is a remarkable thing, right? That the cells in our body, almost all of our cells, have a way to keep track of time, right? To know when it's daytime and when it's nighttime, right? And I mean, this is, when you think about it, it's remarkable, right? It's like having a wristwatch, but inside the cell. And so the issue really is, I mean, how do cells keep track of time using biological molecules, right? They don't have a wristwatch, but they have to have some biological instantiation of a watch. And so this is really an amazing thing, and it's common to many animals. And you all know that you have a clock because you've experienced jet lag, which is what happens when your internal clock is out of sync with the changes that occur in the environment. And we'll talk more about that. So, so circadian clocks are 24-hour biological oscillators, right? The watch is just an oscillator. The hands are going around and around and around. And this watch, this like old school wristwatch here that's mechanical, you know, every 12 hours things repeat, right? But of course, these oscillators, the circadian clocks oscillate with 24-hour periodicity, and we'll talk about that more in a minute. As I said, for the oscillator to be useful, for the timekeeper to be useful, it has to be synchronized with the environment, right? So the cell has to know when it's morning and when it's night and not confuse the two. And we'll talk about how that happens. And the most special property of these circadian clocks is that they can run even in the absence of external cues. And so what I mean is you can put cells in constant environmental conditions, so constant light, and they keep track of time without any cues coming from the environment. Okay, so we call that property the ability to free run, and that's a hallmark of a true circadian clock, right? And this is in contrast to something that's not a real circadian clock, which some organisms have, that requires input from the environment to keep running. Okay, so that's more like an hourglass, right? Where every 12 hours, let's say, you'd have to turn the hourglass over. So real circadian clocks run without any interference or input from the environment. So the clocks are found in, as I've already alluded to, all kinds of animals, including humans, rodents, fruit flies, plants, some fungi, and also some bacteria, which is what I'm going to tell you about. In terms of humans and animals, they control all kinds of physiology that's important, and I think all of you, again, know this because you've experienced it. They control our sleep-wake cycle, they control how alert we are, they control and influence our patterns of digestion, and for example, things like body temperature, which also fluctuates with circadian pattern. And this word circadian, of course, means approximately 1 day. So first, a little bit of definition. So this is just a schematic that represents some fictitious output from the clock. This could be anything, like some signal that controls body temperature, right? And it oscillates with circadian periodicity. And so the period here is just defined as the peak to peak or trough to trough time. And in this case, it's 24 hours. Again, this word circadian, approximately 1 day. So that's the period, the peak to peak time. And the other term I'll use is the phase. So here I've just diagrammed a second oscillator with this dashed line that's offset from the first one, the solid line. And so this offset here is referred to as a difference in phase, right? And in this example, the difference in phase is 12 hours, right? Both of these oscillators are oscillating with 24-hour periodicity, right? For this dashed line 2, the peak-to-peak distance is 24 hours. Okay, so phase and period, those are the only two kind of technical terms I'm going to use here. You may also be aware that the Nobel Prize this year was awarded for the study of circadian clocks in the fruit fly Drosophila melanogaster, where Jeffrey Hall and Michael Rosbash, working at Brandeis, and Michael Young at Rockefeller, identified the first genes that encode components of the circadian clock in the fruit fly. And most importantly, they proposed a mechanism by which these gene products, the proteins, might produce oscillations. All right, and we're going to talk about that model and then talk more about these bacteria that I was telling you about. Okay, so what Rosbash, Hall, and Young did is to propose a model for how you might get these oscillations, okay, inside of cells that are used to keep track of time. And the model is referred to as a transcription translation oscillator. And it's some fancy words for a rather simple mechanism. So here if we have a gene, let's say a clock gene, that's in the nucleus, and here this arrow refers to the start of transcription, the line represents, and the box represent the DNA, right? So this gene is transcribed, and the messenger RNA that's produced during transcription is then exported from the nucleus, like essentially all messenger RNAs. And then it's translated in the cytoplasm to make the corresponding protein, let's say some clock protein called X. Now what happens is it takes some time, but this protein accumulates, right? It's being produced and accumulates. And in this model, it's somehow modified in a way that affects its activity, right? This could be post-translational modification, which I think you've learned about, things like phosphorylation, but it could also be other modifications. And so this protein X, the little graph here, just accumulates over time, right, through the process of transcription, translation, and then this modification. And then what happens in this model is that the protein is imported into the nucleus, where it represses the transcription of its own gene. All right, so this is just a feedback loop, right? The protein's made, it accumulates, it's modified, imported in the nucleus, and now it turns off its own transcription. And so what happens is you get accumulation of the protein through this normal process of biogenesis, essentially. And then you have the protein levels decline because transcription's been shut off, so no more messenger RNA is made, and so no more protein is produced. All right, so you get this sort of parabolic shape here. And of course, the whole thing can start over again if you propose, and this is what Rosbash, Hall, and Young proposed, that this protein X is then degraded. And now if it's degraded, the whole cycle can start again, right? The gene can be transcribed because it's not being repressed, and so on. Okay, so this is the transcription translation oscillator, and the reason it's called this is sort of evident, that the oscillations require transcription and translation, right? If you block transcription or translation, you can't get oscillations. Okay, so that's the sort of idea that they were awarded the Nobel Prize for. All right, so some of the big questions in the field, the circadian field, are what are the parts of the clock, right? So if we have a wristwatch here, this isn't a wristwatch, this is some alarm clock, but if we opened up a watch, you'd see something complicated like this. And to understand how the clock works, we need to know what the parts are, right? What's under the hood here inside the clock. Something a little more complicated is we need to understand how information about time is read out. So with this clock here, or the wristwatch on my hand, it's really simple, right? The thing has little hands, and we just read out time directly on the dial. But the cell can't do this. It doesn't have, this sort of mechanical mechanism. So how is time read out? How is it encoded inside a cell, right? How do you use biological components to keep track of and then read out time? And we'll talk about this. And finally, and I think one of the most peculiar things, is how are oscillations generated? I told you about this transcription translation oscillator idea. And that topology, that architecture, the structure that I showed you, is thought to be common to all circadian clocks. But I'm going to show you a system where oscillations are generated in a very different mechanism. Okay, so the organism we study is this crazy one called Cyanobacteria. They look like this when they're grown in a flask, this beautiful green color. And this is because they use photosynthesis to get essentially all their energy. And they have pigments that look green under ambient light. Those pigments are critical for photosynthesis. So they're these rod-shaped bacteria, single cells. They live in water, fresh water and salt water. They account for a substantial fraction of the photosynthetic activity in the Earth's oceans, so on this planet. They're the ancestors of chloroplasts in modern-day plants. Many cyanobacteria, in fact most, have a circadian clock, and we'll talk about why, and I'll talk more about this in the afternoon talk. And so why do we study this crazy organism? I mean, why not study humans? Well, the answer really is that this is a simple organism that has very powerful genetic tools. And I won't say a lot about this, but you'll get the gist of it from what I had to say. Okay, so why do cyanobacteria need a clock? And again, we'll talk more about this. I'll tell you part of the answer. Many cyanobacteria do two biochemical processes that turn out to be biochemically incompatible. So I've already told you that these bacteria are obligate phototrophs. That means they require sunlight and photosynthesis to produce ATP. And of course, photosynthesis requires molecular oxygen. But many of the cyanobacteria also perform nitrogen fixation. And this process turns out to be exquisitely oxygen sensitive. So if you have an oxygen sensitive process and another one that requires oxygen, this is what I mean by biochemical incompatibility, it's a problem for the organism, right? And the organism, there are many different solutions, and different cyanobacteria use different solutions. And what I mean by that is that you could have these things spatially separated, right? You could have an organelle-like structure where nitrogen fixation takes place in a separate place for photosynthesis. And that is one solution that some cyanobacteria use. But the other solution is to separate them temporally, right? To schedule them to take place at different times so they don't interfere with one another. And that's what many cyanobacteria do. They do photosynthesis when photons are available during the day, and they do nitrogen fixation at night. Okay, so one of the coolest things about the cyanobacteria is that we can see circadian rhythms inside single cells. Okay, and I'm going to show you this. And the way we visualize this is really rather straightforward. So we generate what's called a circadian transcriptional reporter gene. Okay, and this is a gene, it's a piece of DNA that produces a fluorescent protein. All right, so the DNA encodes this fluorescent protein. And you probably heard about this, things like green fluorescent protein, right? So, and the gene for the fluorescent protein is controlled by a regulatory region, a promoter is what we say, that is responsive to the circadian clock. Okay, so it's really simple. There's a transcription factor that binds here that's controlled by the circadian clock, and now the transcription of the gene encoding the fluorescent protein is under circadian control. Okay, and so schematically, if we measure fluorescence, and I'm going to show you the real data, what you see is that fluorescence oscillates with circadian periodicity, right, 24-hour periodicity. So this is how we can visualize circadian rhythms in living cells. And that's what's shown on this slide here. So what we do is we can grow bacteria. So here's two bacteria. This is a phase contrast image on the top. And we grow them in these little channels here, all right, so that they grow in a really ordered fashion. These things they're rod-like, I told you before, and they grow just by division. And lengthening and dividing, and you'll see this in the movie down below. So we keep them confined to these little channels so we can easily measure what's going on. And so that's phase contrast above, and now here's the fluorescence image on the bottom. And that's what you'll want to look at. So I'm going to play the movie, and it's sped up 43,000 times. Otherwise, we'd be here for a long, long time. What you'll see is that the cells grow and divide, and look what happens to the fluorescence. Dim and bright and dim and bright, okay? And it turns out that this is oscillation, with 24-hour periodicity. And you see one remarkable thing. The cells are marching essentially in lockstep here, right? Where the intensity is changing, growing, and diminishing in the same way in all the cells in the field. Okay, so that's pretty cool. We can then measure the fluorescence in individual cells to tell us about what's going on with the clock inside these cells. And that's exactly what we do. And so the first question I posed is, what are the parts of the clock? And that was the first thing that people answered about this system that I'm talking about. And as I said, inside a wristwatch, if you opened it up, not the new digital kind, but these old ones, you'd see all these little springs and wheels, and each of them have a specific function. And you'd ask whether each of these pieces was important by just removing one, right? That's like sort of conceptually the simple thing you do. If I wanted to know whether this piece was important, I'd take it out and I'd see what happened to the ability of this thing to keep time. And that's the same thing that we do in biology to identify the parts of the clock. We break apart inside the cell, and then we ask what happens to the function that we're interested in this case, the clock function. And so that's what the Kondo lab did in Japan, now about 30 years ago. They looked for cyanobacterial cells. They mutated the cells and looked for ones that no longer show oscillations, right? And they used the kind of reporter I described on the previous slide. So here's what we call wild type. That means like normal, not mutated, right? They exhibit these 24-hour oscillations. And Kondo looked for mutants that no longer exhibited oscillations. All right, and so once you have the mutants, you have to identify the genes that are mutated, and we'll skip that part. And nowadays, that's actually very easy with new sequencing techniques. But you identify the genes that are mutated, right? That's equivalent to the parts inside the clock. And then what you do is study the proteins that are encoded by those genes, right? So this is all about the parts. And it turns out this clock is made up of 3 parts, chi A, chi B, and chi C, that are depicted schematically here. Chi meaning clock or time in Japanese. So chi A and chi B will come to. Chi C is a hexameric protein. That's why I've drawn these 6 blue balls here. It's a hexameric protein, and it's perhaps the most interesting of the 3 proteins, and we'll get to that in the next slide. So those are the 3 parts, and if you mutate any of them, the clock doesn't work anymore. There are no oscillations. So these are necessary to generate circadian oscillations in that bacteria I told you about. And now we want to understand, what these proteins do and characterize them biochemically. So chi C, as I said, is perhaps the most interesting of the 3 proteins. Turns out it's an enzyme. We'll get to that in a minute. And it is also rhythmically phosphorylated in vivo. Okay, so I'll just remind you that phosphorylation is a covalent modification where the terminal phosphate on ATP is used to covalently modify, in this case, serine and threonine residues in the polypeptide chain of this protein chi C. And so I'll refer to as chi C as either P chi C, that's phosphorylated chi C, or unphosphorylated. And schematically, the black ball just represents phosphorylation. Okay, so, and I'll use this sort of these little pictures throughout. So Chi-C phosphorylation oscillates with circadian periodicity. This is a schematic shown here, but the data really looks very similar to this. And as I said, Chi-C itself has enzymatic activity. And it turns out that the phosphates are put on Chi-C by Chi-C itself, probably in trans, meaning a different molecule of Chi-C than the one being phosphorylated performs this enzymatic activity. So Chi-C has this autokinase activity, we like to say, and it also has phosphatase activity, meaning it can remove the phosphate, the black ball, to produce unphosphorylated Chi-C. So it has these two enzymatic activities embedded in the same protein. All right, and the first big surprise here is that inside the cell, these oscillations in Chi-C phosphorylation don't require transcription or translation. Okay, so this was a huge surprise at the time because the sort of dogma in the field was that all circadian oscillations are generated via this common mechanism of transcription, translation, oscillation, right, that I told you about. So if you block transcription or translation in these cells, phosphorylation of Chi-C continues unabated in this circadian manner. All right, so that's peculiar. But maybe you'd say, well, maybe transcription or translation is not completely blocked, or there's something more complicated going on. But the real experiment here, and I think the most surprising thing in this whole field, is one that Kondo did to show that you could take purified proteins, Chi-A, Chi-B, and Chi-C. Now he purified these proteins expressed them and purified them from E. coli, a different bacterium, and mixed them together with magnesium ions and ATP in a test tube, and nothing else. And he reconstituted oscillations in the phosphorylation state of Chi-C. So this is crazy, right? You mix 3 proteins together with ATP, and you get oscillations that persist indefinitely as long as ATP is present. Okay, so that's like not what you expect, right? When you do biochemistry and mix things together, this is not what you normally see. And it was this is a really important result because it demonstrates that only 3 purified proteins are needed to get these oscillations, and that the oscillations must arise from a mechanism that is post-translational, right? It doesn't require translation. It happens after translation of the polypeptides, right? Because these things are already translated and folded and whatnot, these 3 proteins. So this was a really important result, and this is where my lab got interested in this problem, and we wanted to understand how this happens, okay? And I'll say more about that in the next few slides. So first I need to just complete this, this information about the biochemistry of the clock proteins. So I've told you that Chi-C oscillates between this unphosphorylated state without the black balls and the phosphorylated state. And that this oscillation is controlled. These phosphates are put on by Chi-C. It's a kinase. That means puts the phosphates on. And removed by Chi-C, this phosphatase activity. Okay? So now the other 2 proteins play very important roles in controlling whether Chi-C is putting on phosphates or taking them off. And it turns out that when Chi-A is present, it stimulates Chi-C kinase activity. All right, so I mean, if you just mix Chi-A together with Chi-C, Chi-C becomes nearly completely phosphorylated. All right, so Chi-A somehow interacts with Chi-C to promote phosphorylation. And this other protein, Chi-B, does something a little more complicated. It stops Chi-C phosphorylation by inactivating Chi-A. All right, so if you mix Chi-B with Chi-C alone, well, Chi-A isn't present, so you don't get phosphorylation of Chi-C, and Chi-C is mostly in this unphosphorylated state. If you mix Chi-B with Chi-C alone, I told you mix Chi-A and Chi-C, you get the phosphorylated state, and if you mix the three proteins, you get dephosphorylation of Chi-C because you block this Chi-A autophosphorylation activity. And we'll talk more about this, but now you can begin to see how you might get oscillations between these two forms of the protein. All right, so one of the first big conceptual questions we tried to answer is, how is information about time read out? And implicit in what I've been saying is that time is read out in the phosphorylation state of Chi-C, right? If that's what's oscillating, maybe that's how time is read out. And it can't be that simple, right? Because, for example, if we look at these 2 points in circadian time, the 2 red balls here, what you see is that the level of Chi-C phosphorylation passes through the same point. I've used 50% phosphorylated Chi-C as an example here, at 2 points in a single circadian cycle, right? It passes through this first point in the rising phase of phosphorylation, when phosphorylation is increasing. And it passes through this second point, the same 50% point, when phosphorylation is decreasing, the falling phase. So you can't use total phosphorylation on Chi-C as the way to read out time, or you would never know if you're in the rising phase or the falling phase. It's just like this wristwatch, right? Now it says whatever, some 20 to 10. I don't know if it's AM or PM from this watch. And the same is true here. So this can't be the way the cell reads out time. But I'll just tell you and explain this in the next few slides that it's certainly related to the phosphorylation of Chi-C. All right, so the devil is in the details here. And it turns out that Chi-C has 2 amino acids that get phosphorylated. Okay, I've drawn it as one black ball and just total Chi-C that's phosphorylated. But it turns out there are 2 different amino acids that are phosphorylated. I know you already learned about amino acids. So S is serine and T is threonine. And I'll now color code them as I have here. So serine is red and threonine is green. You don't have to remember, but just remember they're different. And because there are 2 amino acids that can be phosphorylated in each polypeptide of Chi-C, there are at least 4 possible phosphorylated forms of Chi-C. All the way on the left is the unphosphorylated one. All the way on the right is completely phosphorylated on both serine and threonine. And then you could be phosphorylated on serine alone or threonine alone. So 4 possible phosphorylated forms. And it turns out that these 4 forms oscillate with circadian periodicity. So here I'm showing one circadian cycle, 24 hours. And in black, this is just what we've been looking at, the total amount of phosphorylated Chi-C, right, without looking at the individual sites. And now we see the 3 different forms that I referred to on the previous slide, right? The green, the blue, and the red. And they all oscillate with circadian periodicity, right? The peak to peak or trough to trough ratio is about 24 hours for all of them. But the phases are different. Remember, the phase is just this offset, this shift of these curves here. And so I already said that, total phosphorylation. So the green one is phosphorylated on threonine only. The blue one is phosphorylated on both sites. And the red one is phosphorylated on serine only. And you see something peculiar happens. The threonine one accumulates first, then the doubly phosphorylated species. And then only much later does the abundance of this red species, the serine only, peak, right? This thing peaks in the declining phase of phosphorylation. And now you can guess how time is read out. So time is stored in the relative amounts of the different phosphorylated forms of Chi-C, right? And this is easy to see. Here we look at these 2 points in the oscillation. Remember, they're about 50% total phosphorylation of Chi-C. But if we look under the hood and ask which of the phosphorylated forms of Chi-C are present, you see that they're different at these 2 points in time. In the rising phase of the clock, when phosphorylation is increasing, you have most amount of the green species and equal amounts of red and blue. And in the falling phase of the clock, the red species predominates. All right, so now it turns out this is how the cell keeps track of time. When this threonine phosphorylated form of Chi-C is most abundant, it knows it's in the rising phase during the day. And when the serine phosphorylated form of Chi-C is most abundant, it knows it's in the falling phase. So that's how time is read out inside the cell. It's read out with the cell's ability to measure the relative amounts of the different phosphorylated forms of Chi-C. And the way the cell does this, I won't say much more about this, is using proteins that interact specifically with the different phosphorylated forms of the protein. Okay, so now I want to turn to this issue of how are oscillations generated, right? And in a watch, we understand this well. But in this system, it's really not intuitive. Okay, this isn't something we could figure out just by thinking about it. So this just restates the problem. How do you get stable oscillations? So I've told you that Chi-C oscillates between this unphosphorylated and different phosphorylated forms, and that yellow protein Chi-A promotes phosphorylation, and the purple protein is Chi-B that promotes this dephosphorylation. And the issue is really why you get oscillations instead of the reaction just going to a steady state. So you all know that if you mixed, so there are essentially 2 opposing biochemical reactions here, right? One of them is putting on the phosphate and the other is removing the phosphate. And if you mixed together enzymes that put on phosphate and remove phosphate with a substrate in a test tube, you sure as heck would not get this oscillations, right? You get something like this, some approach to a steady state. And intuitively, I think you know this is true. This is like if you have a bathtub and you're both putting in water and taking out water, the level of the water in the bathtub, the steady state level, so that means unchanging over time, is just determined by the relative rates of how fast you put it in and how fast you're taking it out, right? If you're putting it in faster than you're taking it out, the level will rise to a certain point, and the converse is also true. But you won't see oscillations like this. All right, so this is a very peculiar phenomenon here. And it turns out that in order to get these oscillations, you need a time delay between phosphorylation and dephosphorylation. This is true of essentially all oscillating systems. So in the transcription translation oscillator, the time delay came from the time it takes to accumulate the protein in the cytoplasm and for it to be modified before it then goes into the nucleus and turns off its own expression. All right, here we need a time delay between these two parts of the reaction, phosphorylation and dephosphorylation, to get oscillations. And you could imagine, again with the bathtub analogy, if you periodically plug the bathtub, right, you'll see the water will rise when it's being filled, and if you remove the plug, it will fall again to the steady state level. And so you can get oscillations. That's this time delay business. All right, and the second thing that you need that is really actually the most interesting part here is that you have to have a way to synchronize the molecules, right? Almost, let's just say all the molecules in the test tube, and there are billions of molecules, have to be marching in near lockstep, right? Even if individual molecules are oscillating, if they're oscillating out of phase, right, you're just going to see this average behavior. So to get these oscillations, nearly all the molecules have to be increasing in phosphorylation here and then decreasing here. How the heck can you do that, right? How are you going to coordinate all the molecules in the test tube? All right, so I'll tell you the answer to both of these and how we figured it out. So the way we figured it out is by studying reactions that contain subsets of the clock proteins. This isn't complicated. We purify the proteins and we can mix them together in different combinations, and that's what we did. Chi-C alone, I've already told you a little bit about this, and then Chi-C with Chi-A and Chi-C with Chi-B. And I'm not going to go through any of the details. I'm just going to tell you what we learned from these reactions. So what we learned is that first, and this is really interesting too, the phosphorylation and dephosphorylation reactions are really slow. So there's something else peculiar here that I haven't brought up, and that is that the period is 24 hours. So how do you get such a long period from biochemistry that I think you all know typical reaction rates are like milliseconds and seconds, right? They're really fast. How can you get a period that long from biochemistry that's so normally so fast? And I'll just tell you, the answer is that it's not fast in this case. These reactions of putting on and taking off the phosphates are really slow. And that is some intrinsic property of Chi-C, meaning that's just how it is. It's a lousy enzyme, effectively. All right, so that's one of the things we learned. And the other thing we learned is that there's an ordered phosphorylation. And you could glean that from the slide that I told you, how's time read out. It's read out by these different by measuring the different phosphorylated forms of Chi-C. So the reason the phosphorylated forms of Chi-C appear at different times is because there's this ordered phosphorylation, and this is really simple. So what happens is, if you start with an unphosphorylated molecule of Chi-C, you always put the first phosphate on the threonine, not on the serine ever. Unphosphorylated, when you put the first phosphate on, it goes on to the threonine to make this green species. And that's why in the rising phase of the clock, the most abundant species, and the way the cell knows it's in the rising phase of the clock during the day, is because T-Chi-C is present. All right? And this is just, again, an intrinsic property of the enzyme. It likes to put it on the threonine. That's its preferred site. But now once the phosphate's already on the threonine, and Chi-A is active, it's promoting kinase activity, the threonine is used up. And so now it has to put the phosphate on the less preferred site, which is the serine, to make this doubly phosphorylated species. Right? So during this part of the reaction, Chi-A is active because these are both phosphorylation reactions, right? First threonine phosphorylation, then the serine phosphorylation is added to a species that already had the threonine phosphorylated. And now the thing has all the phosphorylation sites taken up, and there's only one way to go. You got to remove the phosphates because there are no more sites to be phosphorylated. And now there's an order here. The threonine is always removed first. See, this enzyme likes that threonine, and it's removed so it forms this species, the red one, that has a phosphate only on serine. So this thing comes only from dephosphorylation of the fully phosphorylated species. And then, of course, this phosphate is removed from the serine to make the unphosphorylated form. So there's a cycle here, right? And it's just, that comes from the properties of this enzyme. It likes to put the phosphate on the threonine first and take it off that amino acid first as well. And so now you can see how we get a delay between phosphorylation and dephosphorylation. That comes from the fact that this enzyme has this preference to put the phosphate on threonine and remove it first. It creates this cycle where the phosphorylation events, shown in this pink color, are temporally separated. They're separated in time from the dephosphorylation events shown in the blue box, right? And that happens just because of this preference the enzyme has. All right, so now how you get all the molecules to march in lockstep. And it turns out that this comes from feedback. So like the feedback in the transcription translation oscillator, but a little more complicated here. So I've told you that in the rising phase of the clock, right, Chi-A is promoting Chi-C phosphorylation. So phosphorylation's increasing. And the enzyme wants to put the green ball on the threonine first, and then it puts the other phosphate onto the serine to generate the fully phosphorylated species. And now there's only one way to go. I've told you, down, right? Because there are no more phosphorylation sites. And so the enzyme starts dephosphorylating, and it prefers to remove the green ball first, the threonine, to make this red species, the serine phosphorylated form. Now this thing is the special form, and it's the key to how the molecules march in lockstep. And what it does is, and then eventually, I already said that, S gets dephosphorylated to U. What the serine phosphorylated form does is to bind this protein Chi-B. It physically interacts with the protein. And then this complex of the red species of Chi-C bound to Chi-B soaks up all the Chi-A. And now what happens? What happens if Chi-A is gone from the reaction? It can't phosphorylate, so everything then dephosphorylates, right? These arrows, it goes down. And that's how the molecules are kept in lockstep. The way to think about this is that all the molecules of Chi-C are in a bath of Chi-A. Chi-A is promoting phosphorylation. And so if you control the activity of Chi-A, you control what Chi-C does, right? So if Chi-A is allowed to be active, Chi-C phosphorylates. And if the bath of Chi-A is inactivated by this mechanism I've shown you here, then all the molecules of Chi-C dephosphorylate. And that's how they're kept in lockstep. All right, so I'll finish this part and just remind you that, so the answers to these big questions, the parts of the clock, Chi-A, Chi-B, and Chi-C, this came from genetics from the Kondo lab. Time is read out in the relative abundance of these different phosphorylated forms of Chi-C, the green one during the day, the red one during the night. And oscillations are generated by this remarkable mechanism that I've diagrammed schematically here, where this special phosphorylated form of Chi-C that happens only during the night, binds to the Chi-B protein, and that complex then inactivates all the Chi-A that is trying to promote phosphorylation of Chi-C. Okay, so there are a lot of questions left, and I think we have time for one more. And what I'll tell you about is this process. So I've told you about the core oscillator, right? This is the timekeeping mechanism, how you get oscillations, a little bit about why it's 24 hours, what the parts are. And that's what we just talked about. What I want to talk about now is how the clock is coordinated with the changes in the environment. So the clock evolved to take advantage of predictable changes in the environment, right? The sun rising at a certain time and setting at a different time. That occurs with certain periodicity, and the clock in these organisms evolved to exploit that so that the organism can schedule physiology to happen at the right time, right? If you're this photosynthetic bacterium, you want to make the photosynthetic proteins at dawn, right? And you don't need them at night. So we want to talk about how the clock takes in information from the environment and becomes synchronized to that environment. We don't have time to talk about this downstream part, which is also interesting, what the clock does. And I'll say more about this afternoon. So we're going to talk about that. And this is a process called entrainment. And I wanted to talk about this next because we can all relate to this. This is when things aren't entrained is when you have jet lag, right? You go from one time zone where your clock inside your cells is in sync with the changes in the environment, light-dark cycle, and you go to like Asia, where it's 12 hours off. And in order to, and you feel jet lag because your internal clock is totally out of sync with the clock, the environmental changes. And in order to get over jet lag, that's jet lag, your clock has to adjust. I mean, the environment doesn't adjust. Your clock has to adjust, and that takes days. That's why you feel so bad. And if you don't go as far different in time, you feel less worse. But your clock must adjust. All right? And that adjustment is a process called entrainment. Entraining meaning the clock has to be entrained to the environment, has to be synchronized with the environment. And you probably know that there are special cues for the circadian clock, things like light and food you probably know about. And that's why when you go to another time zone, they tell you to go outside and experience the normal light-dark cycle in the new time zone, eat on the new time zone. So food is an important cue in animals. And it turns out temperature is also a cue. And all these cues can be interpreted by the oscillator, the circadian clock, the timekeeping mechanism, and change its phase without changing the period, just the phase, right? Just this offset. And this happens in all organisms that have a circadian clock. It's a universal property of all circadian clocks. And I'm going to tell you how this works in the bacterium, the cyanobacteria that we study. It's really not understood how it works in any other system. Okay, so I've said in vivo the clock synchronizes to light-dark cues so that the peak level of phosphorylated Chi-C, I've sort of said this occurs essentially at dusk, right when nighttime starts, shortly after dusk. And the way we can measure this entrainment property, the ability to shift the phase, is called phase response analysis. And this is like some fancy word for something that's not that complicated. We grow cells in constant conditions, and then we apply a perturbation. We turn off the lights. That's what we call a dark pulse, turning off the lights. And we apply that perturbation, we turn off the lights for a period of time, and then we measure what happens to the clock. So this is a very simple experiment. So here we're measuring, it says bioluminescence, but this is just this transcriptional reporter we've been talking about, okay? And it oscillates, now here's real data, oscillates with circadian periodicity. And now what we do is to apply right here, in the rising phase of the clock, we apply a dark pulse. Just turn off the lights for a few hours. And then we measure what happens. So in black is what happens if there's no dark pulse. And in blue is what happens if there's a dark pulse. So this is remarkable, right? The phase is shifted after the dark pulse, right? It peaks earlier. That's a phase shift. Okay, so that's cool. So how does the clock do this? How does the clock interpret the dark pulse and change its phase without changing the period? But there's something else remarkable, and that is if you apply the dark pulse in the falling phase of the clock. So that's the same length dark pulse, the same perturbation. You get the red curve, essentially no change in phase. Yeah?
Sorry, you don't have any
Points.
line?
Yes, yes. That's very perceptive. So the reason is, I'll just tell you the reason, because that is very perceptive. In the dark, transcription and translation are shut off in this organism. Okay, and so we can't measure this reporter. And we can talk more about that later if you have more questions. But that's why the points are missing only in the samples where we applied the dark pulse. It's because we can't measure then. The reporter doesn't faithfully report on the circadian clock. All right, so how is information about light, in this case dark, communicated to the oscillator? And I'm just going to cut to the chase and tell you that there are, and we knew this, that there are substantial metabolic effects of darkness. Right? I told you that bacteria is an obligate phototroph. What this means is that it requires photosynthesis to generate ATP from light. And so when there's no light, it can't do photosynthesis. And what happens during the dark pulse, independent of the time at which the dark pulse is applied, whether it's in the rising phase or the falling phase of the oscillation, ATP levels drop. And this isn't surprising. The lights are off, it can't make ATP, and it's using ATP. All right, so what we want to know, so it's really this charge of the adenine nucleotide pool, which is the ratio, the percent of adenine nucleotides that are in the triphosphate form. But don't worry about that. I'm just going to say ATP. So how does a change in this ATP to ADP ratio cause a phase shift? Does it, and how? If so, how? And the answer, of course, is yes, or I wouldn't write this. And why is the oscillator most sensitive in the rising phase? Right? I showed you that during the rising phase, if you apply the dark pulse, you get a phase shift, and in the falling phase, not. So why? All right, so this ATP to ADP ratio, not surprising that it affects the clock, because I've told you Chi-C is an enzyme that uses ATP as a substrate to put the phosphates on itself, right? And we asked whether this balance of phosphorylation and dephosphorylation is altered by changes in the relative ratios of ATP to ADP. And the way we did this is using a biochemical, the in vitro system. Now these are all purified proteins. There's no cells, there's no other proteins, just Chi-A, Chi-B, and Chi-C. And we're going to change the ratio of ADP to ATP in the reaction. And this is, what we're doing is trying to mimic this. This is in vivo. The ATP levels are decreasing in the dark, and we want to mimic that in the test tube. So they decrease during the whole time the lights are off, and then they rapidly increase. So we want to mimic that in the test tube, and that's what we do here. And the way we do it is using a biochemical trick. Well, the first part is not really a trick. We just add in. So there's ATP in the reaction. We add in ADP so that now there's equal amounts of ATP and ADP, right? So we reduce the ratio of ATP to ADP by just adding in adenosine diphosphate. This is trivial. Here's the more interesting part. Now we have to restore ATP levels because that's what happens at the end of the dark pulse. And we do it using an enzyme, pyruvate kinase, that can use a high-energy phosphate donor, phosphoenol pyruvate, to essentially phosphorylate ADP to produce ATP. And this reaction goes to completion in about an hour. So here we add in ADP, and that mimics what happens in the dark pulse. And then we restore ATP levels at the end of the dark pulse using this trick by adding in pyruvate kinase, which produces ATP from the ADP. And now you see what happens in the test tube, right? And now here all the points are here because we measure directly phosphorylated Chi-C. And in the black is what happens when there's no perturbation, and in the blue is what happens when we apply this. We mimic the effects of the dark pulse by changing adenine nucleotide levels. And remarkably, if we do the same experiment in the falling phase of phosphorylation, there's essentially no effect on the phase. So this really is striking evidence that changes in nucleotide levels are sufficient to cause a change in phase here. And that really strongly suggests in vivo that this is the mechanism by which changes in light intensity cause a change in the phase. All right, so now to this issue of, and we can skip this. Now to this issue of what's the mechanism? How does changing this ratio cause a change in phase? And again, we turn to these partial reactions, mixing subsets of the clock proteins together. And I'll just tell you the only important result we got, and that was when we changed the ratio of ATP to ADP in this reaction in which we mix Chi-A, remember, that promotes the kinase activity of Chi-C, with Chi-C. And what you see is that as you change the ratio of ATP to ADP to have more ADP, you see the rate of phosphorylation of Chi-C gets lower and lower, right? So it turns out that Chi-C activity is competitively inhibited by ADP. I think you learned about enzymes. So ADP and ATP compete for the same site, the active site of Chi-C. And so if there's a lot of ADP present, which can't be used to put phosphates on Chi-C, then the rate of phosphorylation decreases. And we can analyze these little curves to demonstrate that these two nucleotides, ATP and ADP, have about equal apparent affinity for the enzyme. And so now we can understand what's going on. So I've told you that Chi-C alternates between this unphosphorylated and phosphorylated form. It oscillates. And that Chi-A is promoting phosphorylation and Chi-B is inactivating Chi-A to trigger dephosphorylation. And now what's happening in the dark pulse, when the lights are turned off, the cells can't make ATP by photosynthesis. They utilize ATP, and therefore the ratio of ADP to ATP increases. And when this increases both in the cell and in vivo and in vitro, what happens is that this causes a reduction in the rate of phosphorylation of Chi-C. And this is why changing the adenine nucleotide levels or applying the dark pulse only has an effect in the rising phase of the clock, when phosphorylation is increasing. That is the only time that Chi-A is active, right? So I'm going to stop there because I think I don't have enough time for the last thing. And also, I want to give you time to ask any questions you might have and not run over. So Don't be shy. Any questions? Anything? Yeah. Awesome.
So you mentioned how there's like at least 4 stages of UTSTS.
Yes.
At least 4. What does that mean?
Yeah. No, yeah. I was careful because every time I look at this, let's see. Let's go. Oh. Yeah. Okay. So, because theoretically, it's possible that there are more. And in reality, there may be more because Chi-C is a hexamer. And what if, let's say this particular Chi-C, this is one polypeptide of Chi-C. What if it also has a green ball? I made this seem like everything's absolute, like T is always put on before S, but in reality, it's probably not, it can't be totally perfect. So there may be hexamers, for example, that have mixed phosphorylation state of Chi-C. That's really, really hard for us to measure, okay? We can't. So that's why I say what I say. I mean, yeah, there could be, this is probably a simplified and extreme view of what's happening. I suspect in reality it's a little messier than this. It's a good question, though. Yeah.
So I guess it makes a lot of sense for like this organism that is dependent on photosynthesis for like ATP regulation, and its correlation with like blood interaction. But like all animals have circadian rhythms. Do we know
Yes.
about that?
Yeah, this is a great question. So you're right that this is probably, this may be something peculiar to this particular organism. However, there are many links between metabolism in humans and in rodents, metabolism and entrainment of the circadian clock. So but the mechanisms aren't well understood. And whether those mechanisms are really the same, meaning ATP to ADP ratio, I suspect not, but I think conceptually there are likely to be the same kind of conceptual links between metabolism and the function of the circadian clock. But the actual molecular mechanism, just as you're pointing out, is likely to be different. Yeah. Yeah.
In the beginning, you said that one important hallmark of the circadian clock is that it can operate without this light and dark cycle.
Mhm.
But in the case of cyanobacteria, it seems like if you put it out of the cycle, it doesn't really work.
Okay, so it's not, so that's a really good point. But the cyanobacteria, if you keep them in constant light, so darkness is a problem because they can't get any energy. But we'll come to that this afternoon. There are sort of ways to get around that. But constant light, if you put, and in fact, in the lab, this is what we do. We grow the organism with the lights always on. The clock runs indefinitely. Yeah. So that's what I mean by constant light. And it's true of people, too. They've done all kinds of experiments putting people in constant conditions, either darkness or constant light. Yeah. Yeah, the clock runs, and that's true for all true circadian clocks. Yeah. But you're right. In this organism, you can't put them in the dark all the time. That's right. Yeah?
I was curious about the hourglass version of the clock.
Yeah. Yeah. So it turns out there are organisms with an hourglass. It's not a real circadian clock, and it's a relative of this cyanobacteria that I told you about called Prochlorococcus. It's another bacterium. if you put that organism in constant conditions, say constant light, it also does photosynthesis, the circadian rhythms just die out. Instead, if you grow it in oscillating light-dark conditions, they persist. So that is an example. That has an hourglass, which may be, and I'll tell you that the component of that hourglass is chi-C. It doesn't have all the chi proteins. It only has chi-C and one of the other two. I don't remember which. So it may be that the clock evolved, this true clock in the cyanobacteria I told you about, evolved from this hourglass-like mechanism, right, that existed in organisms like Prochlorococcus. Yeah.
And if you were to add a pulse, not in a constant situation, so if there still was differing inputs and you did a pulse, would you expect to see, given that there is chi-C present, some phase shift?
Yes, definitely in that organism as well. Absolutely. Yep. Yeah.
You mentioned how cyanobacteria do nitrogen fixation.
Mm-hmm.
So we can grow them in constant light. Do they still cycle between photosynthesis and nitrogen fixation?
Yes, they do. And that is because the mechanisms that control those processes, they're clock-dependent mechanisms, still operate independent of the environment. That's right. Yep. Yeah.
You mentioned that food, light, and temperature were
Yes.
the main mechanisms that created a phase.
Exactly.
And you mentioned mechanisms for food and light. How does temperature
Yeah,
cause a phase?
that's a great question because in cyanobacteria, it's really a dominant cue. We've used it recently for the reason you alluded to. We have used temperature to cause phase shifts and study the details because with temperature, the reporter, transcription and translation aren't shut off, and we can still measure the reporter, and we don't have those gaps that you pointed out so perceptively. So temperature does cause a phase shift, and we do not understand and haven't really tried to study how that works. But it causes as dramatic a phase shift as what I showed you for ATP and ADP. Yeah, it's a good question. We don't know. Yeah.
So you say that after you phosphorylate everything, the enzyme will remove the T,
Mm-hmm.
and then chi B would block chi A.
Exactly.
But then, like, how does it know to remove the T? Like,
Yeah, no, that's a really good question. How does it know to put the T on first, the phosphate on the T first, and then remove it first? I think we don't know, but that's just presumably the enzyme has an active site that it just prefers that threonine. You know, it can tell the threonine apart from the serine, right? The threonine has this branched side chain, and the serine has no branch. And so presumably it's either directly, it's reading the identity of the amino acid, undoubtedly it's doing that, whether it's serine or threonine. And it prefers threonine. Why? We don't know. And you need very detailed structural analysis to really understand that. And there are structures of these proteins, but we haven't really tried to figure that out. It's a very interesting issue, but we don't know the answer. Yeah. Okay, one more.
Do we know what it is about chi-C that makes it such a slow enzyme?
Oh, yeah. Oh, yeah. So that's really a great question. And no. That's the short answer, no. And there are other super interesting properties because I said that temperature can cause a phase shift. But the other peculiar thing about temperature, and is true of every circadian clock, is that the period is invariant over a huge range of physiological temperature. And those of you from chemistry know that chemical reactions are not temperature invariant. In fact, most of them have significant temperature sensitivity. This period is not temperature sensitive. And so and we know that's an intrinsic property of the Chi-C enzyme, and we don't know where that comes from. Yeah.
Well,
Okay.
thanks very much.
All right. Good.